Dr. Lisa Porter is a director of WESpark Health Institute and a distinguished professor in the Department of Biomedical Sciences at the University of Windsor. She completed her PhD at McMaster University in Hamilton, Ontario, in the Department of Medical Sciences, and was a postdoctoral fellow at the University of California, San Diego, La Jolla, California. She obtained her BSc Honors Biology and Pharmacology co-op program at McMaster University in Hamilton. She was a founding member of the Windsor Cancer Research Group and continues to sit on the executive committee. Dr. Porter is a Canadian Institutes of Health Research University delegate and a member of the College of Reviewers. Please welcome Dr. Lisa Porter.
Thank you for the kind introduction. So today, I don't think my area of cancer biology, I don't think I need to do an introduction to cancer to anyone. I'm pretty sure everyone in this room has been affected by cancer at some point in their life, either directly or indirectly. And the sad fact is that cancer affects nearly half of us will actually be diagnosed with cancer at some point in our life, and a quarter of people will die from the disease. But obviously, I don't want to start our morning out on a negative. So really the point of my talk here today will be to tell you that progress is being made, that about 60% of people diagnosed with cancer today survive their disease, and that progress is happening for those types of cancers that affect our youth and adolescents the most, and that quality of life during treatment continues to improve year after year. My lab focuses on several different types of cancer, breast, liver, brain, and prostate cancer. Today, I'm going to talk to you about the first type of cancer that I started to work on in my lab, and I have a rationale for selecting that. But so I'm going to focus on breast cancer. And we know today that in North America, about 1 in 8 women are affected by breast cancer every year, but globally, about every 30 seconds, someone is being diagnosed with breast cancer. And despite the prevalence, it is the second leading cause of cancer-related deaths among women. So lung is the first now. And that's in part because of the progress that's been made, but we still have work to do. I know this animation isn't working, but I'm a cell biologist, so I like to stare down a microscope and look at how cells grow and divide and all of the different things that cause the cell to do this. And really, there's thousands of events that have to be coordinated just perfectly to ensure that every single cell in your body, which right now, as you're sitting here, cells are continuing to grow and divide. And so those events have to be very perfectly coordinated. And we know that, we know that that's happening because of the events. Some of these things are a little bit off because I work on a Mac. But we know that these events are perfectly coordinated because of the orchestrating center inside your cell, your DNA, which is actually organized. Every single strand of DNA is about 3 billion different chemical pairs that are organized into messages. So we have about 20 to 25,000 of these genes that are organized within your DNA. And those DNA genes do many, many different things. But one of the things they do is make proteins. And I like to consider proteins in your cell to be the workhorses of your cells. They make a cell look as it should look and function as it should function. So every single one of your cells has all of these different things going on, and they're changing and doing what it's doing every single minute. And even more complicated is that all the different cell types in your body have different proteins doing different jobs. And that's why different cells look different and do different jobs, right? So different proteins are doing different jobs at all different times in different cells. So just remember that point, because I'm going to get back to it. Despite cancer being around since literally as long as written record, so at the beginning of written record, we could see pictures of people with tumors, etc. So cancer has been around for a very long time, but it wasn't until 1979, I think a few of us were around at that time, 2 guys, Michael Bishop and Harold Varmus, told us what actually causes cancer to start, and that's changes to this DNA. So when a DNA changes how it looks or its function, the proteins downstream also become changed. And so that was the very first idea that this is actually what's causing cancer to start, 1979. So we know now that because those changes to the DNA, those proteins either start doing too much or too little. And that's going to change the function of the cell. And so one of the driving forces and how these proteins are changing their function causes the hallmarks of cancer to begin. And one of the main hallmarks of cancer is that these proteins, when they're doing their jobs, are going to cause a cell to grow and divide too quickly. I'm going to show this little kind of schematic, and this isn't a cell. It's actually the processes that a cell has to go through. And so those organized thousands and thousands of events that a cell has to do in order to grow and divide are actually orchestrated in a perfectly orchestrated fashion. So they have to go through a segmented sort of events in order for those divisions to happen perfectly. And they're actually timed and throughout this sort of cycle. So in my lab, we look at this kind of cycle, and I can't get this going right, but the cycle and how it's orchestrated perfectly. I guess if I look at the computer, I can do that better. And so we know that in a cancer, that these are now going faster and faster. So these proteins are doing too much. It's going through this cycle faster and faster. And we know, like all of us, if you're doing a job too fast, you're going to make more and more mistakes. So as a cancer does more and more of these events faster and faster, it starts making more and more mistakes. And that's what's allowing it to gather more and more errors. And that's going to progress a cancer to actually continue to grow and divide. So one of the things, even as biologists like me, we're figuring out what's causing cancers to actually become cancer. Fortunately, there were people that were also sort of taking a more of a hammer approach to what's happening with cancer. They were taking thousands of different and hundreds and thousands of different plant and animal extracts. They were putting them on cells, and they were asking, what can we do to cause these cancer cells to die? And somebody mentioned the Dandelion Project. This is kind of the approach that this kind of approach takes. You take everything that we know that could possibly be working, and we know plants are, for example, a very potent process. They have lots of different things in them that can cause cells to behave differently. So they took all of these hundreds and thousands of things and put them on cancer cells. And that was the original origins of some of our chemotherapy drugs. And we know now that these chemotherapy drugs work on different stages of those processes that I study in my lab. So for example, this chemotherapy drug here, camptothecin, cyclophosphamide, can't see that far, works on one stage of the cell cycle, and then and the processes that the cell works on, and these other ones work on different stages. So different chemotherapy drugs work at different stages. These particular chemotherapy drugs that came out of studying hundreds and thousands of these different extracts is something that we call ACT. And for breast cancer, up to 1995, people were starting to study these chemotherapy drugs, putting them on breast cancer cells. They realized that this combination actually stops breast cancer cells from growing and dividing. And so they, different oncologists and whatnot would work with different combinations. And they figured out that this combination works together to cause breast cancer cells to stop growing and dividing. So we call this ACT. I'm going to refer to ACT a lot. So ACT is the common chemotherapy drug used for breast cancer. This is a death rate graph. So basically, you want this death rate to decline. And from 1930 to 1990, the only thing we had for breast cancer patients was surgery. And so the death rate surgeries started back actually in 1700s, eerily before anesthesia. So we were doing surgery for breast cancer for a very long time. So that death rate had not moved, right? It was just at the same. When ACT actually got worked out, it was the first time in history that we actually saw that death curve start to go down. So death rates started to decline because we had a chemotherapy regimen that was actually doing something. But then that was up to about 2015. People like me kept thinking, okay, but can we do better? Right? So we know now that cancer is caused by changes to the DNA. And I told you in that earlier slide that the proteins that work in different cells do different things. So people started thinking, okay, could we actually do something that wasn't just a hammer on all of your cells that's going to cause lots of different cells to have different effects and sort of get lots of side effects, etc. And so is there a way for us to look better at cancer? And so what people figured out was that in breast cancer, there's actually these three antennas on normal breast cells, but they're increased in breast cancer. So these are these hormones. You might have heard of these things before. So progesterone and estrogen, and this one called HER2 new. They figured out that these are high in breast cancers. And so pharmaceutical companies came along, and I was in pharma at that time. And so we were working on a project on this. And pharma then figured out, okay, let's make drugs that block these antennas specifically. And so these then became either hormone interventions or these HER2 new inhibitors could specifically block breast cancer. And at this time, this is a bit blurry, but this is actually a survival curve. So as every line ticks down, a patient dies. So you can see here that in red, this is ACT. So I told you this is already doing better. This is chemotherapy drugs, but you can see that for these patients at about 5 years, about 66% of patients are only surviving. So we've got like a decrease in death. So we know that we're not doing good enough. But with the addition, this is the one of these drugs, both of them gave about a similar curve. With the addition of this more specific therapy, now you see there's a change in that curve. So about 20% more patients are surviving with the addition of this more specific approach. So this was telling us that, okay, this specific approach now again is going to improve patient survival. So we kept looking at things, and this is about the time that I started my lab here in Windsor. And I came from San Diego at the time, and our lab was at that time in the cancer center, and we were on the everyday, people like me having conversations with clinicians and talking about, what do we need to do better. So I reached out to Dr. Carolyn Ham, and she became the first clinician that I met here in Windsor, Essex. And we, started having lunches together, et cetera. And we started talking about the literature of what was happening with breast cancer at the time. And we said, well, we know that these people with the antenna on those breast cancer cells are doing much better. There was an emergence of literature saying that some patients don't have any of those antennae on their cells. And of course, clinicians were still trying to give them the drugs, et cetera. We didn't at that time even have a way to properly screen patients, but things were getting better to figure out which patients had those receptors, which ones did not, and who did respond to them and who didn't. But we figured out that there was a group of patients, and the literature was figuring out that didn't have any of these. So we call these a triple negative population. And this patient population really had no specific therapy for them. The only thing that they had was ACT. What turns out also is that these patients tend to be our youngest population of patients, and it's the most aggressive disease. And so they're the patients that are getting only chemotherapy. And Carolyn said to me, well, there's a few reports in the literature that suggest that maybe this other type of chemotherapy called cisplatin might be doing a little bit better on these patients. And because I had actually studied this cisplatin drug in my PhD, and I look at how these progress through the cell cycle and this process of how these properly orchestrated proteins are working, I said, well, that's interesting because the cisplatin drug works at a completely different step of these phases that cells have to go through that our other ACT drugs do. So it makes sense sort of in a cell biology fashion that these things would work together. So her and I were talking and we said, okay, this would be really cool. So let's combine cisplatin with this other combination of drugs that patients are getting. And so at the time, and so this was sort of our Windsor experience leading up to the study. Again, this is survival curves. So every time the line ticks down, a patient has passed away. And these are looking at different stages, stage 1, 2, 3. So stage 1 is your best case scenario, more patients survive. Stage 3 is your worst case scenario, more patients are dying. But this was sort of our rate. So the average sort of patient survival was around five years or so, especially with stage 3 disease. And so Carolyn and I, at the time, the Windsor Cancer Center Foundation had this Seeds for Hope grant that they had put out. And Carolyn and I got this 2-year grant for $60,000 for us to look at this question, adding this carboplatin to ACT. And so over the next few years, we would do this trial where the patients would come in, they consent to trial, I would take some of their tissue, and then she would administer the ACT plus carboplatin, and we would look at how the patients did. And then in the lab, I would have those tissues and I would get to do a whole bunch of fun things in the lab, looking at what was going on with the patient's tissue. Over the next few years, 90 patients would get enrolled into this trial, all of them triple negatives, our youngest cohorts being around in their 20s, up to 72, our average age was 51. And so then this was some of our early data. We actually have more recent data that Carolyn's putting together, but we would study them up to 10 years. And so we've got these two lines here are historical perspective. As I said, as this is clicking down, this one here is with no chemotherapy. So at that time, if you can believe it, right, we still have data with patients that had no chemotherapy. 1995 is not that long ago. And so you can see this is how those triple negative patients, then we didn't call them, this is historical, all cancers. This was adding in ACT treatment. Again, many of these we didn't even know if they're triple negative, so probably a more optimistic line. But you can see it's down here. This was our first regimen in green. So Carolyn, again, we have to think about timing. And this was another beautiful thing at the time. And, you know, just even now, we're still thinking about combining sort of the knowledge that I have as a basic cell biologist, understanding when and how these drugs work and how we're timing to give these basic chemotherapy type drugs. And this first regimen, you can see we still, initially patients did a little bit better, but then it started to drop off. So that first regimen, we said, okay, we need to fix the timing of it better. And this was the final regimen that she worked out up here. And to date, this is sort of our numbers. So this is historical perspective for stage 1 patients. So this is your best case scenario. But with our new regimen, 100% of stage 1 patients up till 10 years have not passed away. Stage 2 patients, 95% of the patients have not passed away. Stage 3, I can say that our numbers are around 88%. So they were not as statistically significant as other patients. So we didn't see as great a benefit for stage 3. And some patients at about the 8 to 10 year marks started to relapse with stage 3 disease. So now we're saying, okay, can we do even better? Obviously, we've got this improved regimen and we're moving forward into a randomized control trial, which is the next step that you do. Only patients in Windsor right now are getting that trial, right? So patients other places are only getting ACT with triple negative breast cancer. So this shows you already having these types of research in your own center provides better care. But, you know, we can also say, okay, what can we do now for these stage 3 patients that didn't seem to be getting as good a benefit? And so I told you that as patients were coming in and Carolyn was collecting clinical data, I had their tissues in my lab. And there's a lot of different things I can do with this. One of the things I do is take the different patient tissues. We do something that we call micro dissection, so I can look at different parts of the tumor, and I can array them onto different slides, and I can look at what's going on with different proteins within those cells at different stages, etc. And one of the things that we did was I compared the protein, and even Carolyn and I got after the fact for patients that are relapsed, I could compare the proteins of the patients that didn't relapse, the patients at stage 3 disease that responded versus those that didn't respond. And one of the things we found was this protein called SPY1, which seemed to be higher in patients that relapsed versus those that did not relapse. Interestingly, when I looked at what this SPY1 protein did, we found out that this SPY1 protein tends to be able to override drug responses. It's indicative of different types of basic biology being able to cause cells to grow and divide more rapidly. So my lab has really been focusing a lot on these SPY1 proteins, but we wanted to ask, you know, do these proteins drive relapse and aggressive disease to happen? So Carolyn and I applied. We got a $800,000 CIHR grant over 5 years to study these SPY1 proteins, bring in a huge team of researchers from Windsor Regional Hospital to look at these proteins in different breast cancer patients. We implemented the same kind of platform where patients would get ACT plus platinum. This time I would implement what we call a fresh tissue trial, where we would take tissues from patients at surgery, so before and after treatments with the drugs, and I put them in a variety of different types of fun models in the lab, including these fish that are no bigger than my fingernail, mice. We would then implement clinical trials on fish and mice and test all sorts of different things and look at how these proteins are implemented. And just to make a long story short, what we figured out is that, first of all, the application, so this allowed me to even take these tissues and look at the addition of carboplatin in this platinum drugs. We can see that even in the cells of the patients we can show that the addition of that different drug that we added actually decreases, this is living cells, so it actually decreases the survival of those cells. And that if we manipulated this speedy protein in this stage 3 disease, that even with ACT alone, we were able to cause more of these cells to die. So this is, at the time, we don't have a drug against this protein, so I'm using viruses and all sorts of things to try to decrease this protein. But that we could increase the ability of these proteins, of these cells to die. So the next thing I wanted to do, of course, was try to make a drug against this protein so that we can cause better survival. Fortunately, we hired this guy at the University of Windsor, Dr. John Trant. He said he had a slew of chemists looking for things to do. So I said, I got lots of things your students can do. So we put them on this project to develop a drug against the speedy protein. We got funding from the Ontario Institutes of Cancer Research for just over $1 million to look at developing this drug. That currently is in progress, but he has some very good progress working on this. But the idea is that now already in Windsor, we have where previously all these patients would be getting only chemotherapy, ACT. We've now implemented this ACT plus carboplatin, which we know already increases survival rates. But we found a way now to look at patients that have this elevated SPY1 protein. And what we're now doing is saying, okay, for these patients that we know we're not going to survive as well, can we make better drugs and look at how we can better treat these patients? So the future of that would be that for patients that have that high SPY1, that we're going to give them an improved treatment and increase their survival rates even further. And so, you can see that over time, even understanding these general chemotherapy drugs, I said, started to decline that death rate for breast cancer patients. And then the understanding of, and that was just with, sort of hormone and chemotherapy drugs sort of decline that even further. And so our goal now is for us to improve treatment for triple negative, so to make our carbo regimen sort of standard of care, and to continue finding these new targeted therapies so that we can continue to decline that survival curve even further. And I think that one of the things that we really learned through this process is that, it's super important for what I consider discovery researchers like myself to be constantly talking to our medical oncologists and to be implementing sort of what we're doing into the clinic. Not only does it bring new therapies to the clinic, it brings new infrastructure. So with these grants, I've been able to bring in things like PCR machines to look for that speedy protein that they can use for other things, right? We could bring in that could be then used for things like more accurate diagnostics. That's providing our patients with novel treatments before it becomes standard of care. So we don't have to wait 10 years for policy to change. It's driving the continued learning of our skilled trained personnel. Now Carol at Ham doesn't have to go into a room with a 26-year-old and say, I'm sorry, this is all I have for you is this general chemotherapy. She can say, we don't know what your outcome is going to be because we've got these new therapies that we're able to give you. It fuels sort of an engaged environment because now we're engaging all of the different people within the cancer clinic in research and sort of kind of them being involved in pushing things to the new limits. And I think it provides hope for patients. And so we know that this requires huge teams of people, that people do have to work above and beyond. When we ask people to go and do these things, even at the pathology level, for example, or in surgery, they have to not just take the tissue and put it into a bucket of formaldehyde. They have to actually spend a bit more time in surgery, and they're not getting paid to do that, right? They're doing it because we've asked them to be part of this trial. So it takes people working above and beyond to work together to move things so that things are happening at a better scale. So what Carolyn and I then decided to do was to kind of push the curve further. And we had been working with this Windsor Cancer Research Group within Windsor Regional Hospital, but we said that it should really expand beyond just Windsor Regional Hospital and that research should be implemented and we should start engaging all of our different hospitals in research across Windsor Essex. And so that's when we started the WESpark the WESpark Health Institute. So we officially launched in 2020 as the hospital doors were closing for COVID, but it was actually a great time because we were able to actually implement a variety of different COVID trials. And so we actually were involved in a lot of the different COVID screening projects and whatnot But now our partners are Hotel de Grace, Erie Shores Healthcare, Windsor Regional Hospital, St. Clair, and the University of Windsor. They've all joined on to WESpark. This is a little bit about WESpark. So you can look onto our website to learn a little bit about the values, the mission, our strategic priorities, really to grow health research here in Windsor, Essex. Just over our first 3 years, we have 34 core members. Our core members are people that now have national or international funding for health research. So when we started, there was only 3 of us that had national, international health research funding here in Windsor, Essex. Now, actually, there's 37. So the stat is already out of date every time I move here. And just this year alone, those 34 people, and still again growing, brought in just over $17 million just this year in health research funding. So over 3 years, we've seen astronomical growth in health research here in Windsor, Essex. And, you know, I think I look back to really the first health researcher here at the University of Windsor, Dr. Mike Dufresne, who was really instrumental in starting the Seeds for Hope project, which Carolyn and I, it really kicked off our work together. And this was a saying that he had, and it's become our tagline: without research, hope is just a word. And I get all emotional when I say that. But I just want to thank my lab. So this is really all the research that I told you about today was from one person in my lab. So I got this huge team doing other projects as well. But Dr. Brianne Fifield, who really is just a superstar in my lab. And this is the funding that has funded the majority of that work. And of course, thanks to the patients in our trials. Something noted. And that's all. I can ask for questions. Okay. Yeah, maybe.
Take this off so we can see people. Oh yeah, I'll let you do it.
Yeah.
Excuse me, Dr. Porter, the 17.5 million that came in through external grants. Can you talk about who gives you that? I.e., is it big pharma? Is it foundations?
Yep.
And is any of that monetized, i.e., when there's patents that are developed?
So yeah, that 17.5 or whatever is coming from national or international grants, primarily federal grants. So the Canadian Institutes of Health Research is the major pot of that money. So these are federal grant programs that all researchers have the ability to apply to. The beauty is that 17.5 is the actual grant money that's going in to fuel the research and fund students and whatnot to do the work. In addition to that, the CIHR, for example, pays an additional 40% to the University of Windsor to fuel research in general. So that goes into overhead. So, you know, that's only a fraction of the money actually coming to the city. That would go to the university for them to pay for research, sort of, you know, the labs and turn the lights on in our labs and to pay sort of the research administration processes, et cetera. So, you know, one of our big goals is to grow that, you know. I think when I started at the university, I can't remember what the number was, but our total amount of research dollars coming to the university was something like 10 million. Like now the university is at like 34 million, but you can see that health is about half that. So, and that's just from what has been growing over the last few years. Like we've got some amazing hires that have come here to Windsor. And each of those projects, you know, fuels literally, you know, for me, it's hundreds of students in my lab, you know, that have been funded through research. So this is funding our next generation, getting people involved, besides moving forward the sort of needle on what we're doing. Patents and whatnot, so that's not included in that money. For example, somebody like John Trant, he is a major driver of patent money, etc. Patents actually have a bad rep. Patents are an amazing thing. So researchers need to put more patents down on things because it actually allows you freedom to keep, you know, sort of making sure that what your discovery and that you've put money. So for example, all of my mouse models, I patent so that I have the ability to make those freely available to anybody and to be able to make sure that, you know, that science can be open, etc. So there's a variety of different important pieces to patent funding. And I think that we need to, in Canada, bring a lot more industry and biotech, these kinds of things to Canada. So Canada putting a stake on science is super important.
Thank you.
Yeah, that gives.
That's a really interesting presentation.
No, thanks.
I mean, I'm not a medical person and I understand.
Okay, good. That's-
But
yeah,
so it's interesting to hear the excitement with which you're progressing through this. Also some dates, like it's surprising to understand that prior to, it took to 1979-80 for us to understand what was happening.
Yeah.
And then thank you very much for having lunch with Ms. Pam and Dr. Ham
Yeah.
to come up with all these ideas.
Mhm.
It's exciting. Just a very small question.
Yep.
We have billions of cells in us.
We do. Yeah.
How do the proteins, or how does a cancer cell start?
Mhm.
Do we all have them?
Yeah.
Or
Yeah. No, that's a great question. And I mean, I think this is one of the things that people always ask me, especially in the brain field where, like, typically brain cancers affect younger populations. I'll go into a room of patients and parents, usually, because it's lots of these are kids. And they'll be like, what did I do wrong? Also, so that's like a big, so there's the way that these initial changes start. First of all, do we all have them in us? Yes. So your cells right now, as you're sitting here, those changes are happening to your cells. As your cells are going through those normal events, mistakes happen. They happen just sporadically. We know that. It's why we're upright with opposable thumbs. We evolve. So those changes happen sometimes for the better. But one of the things we have evolved to have is what we call repair proteins. So some of those proteins that are doing jobs in your cells are to find the mistakes and fix them. So your cells are constantly monitoring what's going on. One of the things we know is that as we age, those repair proteins go down. So as we age, the chance of a mistake slipping by increases. That's why the #1 risk factor for getting cancer is aging, right? As we age, our chances of getting cancer increase. If we've exhausted those repair proteins, so it's one of the talks I give for high schools about smoking. We know that as you smoke, for example, that is an environmental cause that can cause damage to those cells. When you're young, you're going to have those repair proteins. They're going to be working overtime. They're going to be fixing those damage. So you're not going to get cancer right away, probably, if you're smoking, when you're young. But every single time that you smoke, there's an increased chance of the error happening. And as you age, if you've exhausted those repair proteins, the chance of errors happening earlier are going to happen. So so every single thing we can do to kind of help to protect ourselves is like wearing a seatbelt in a car, right? So, you're helping to protect yourself. But the fact of the matter is that the sheer random chance of an error happening is there. In fact, up to 2/3 of some cancers are completely spontaneous. There's nothing we can actually do. And that's a fact. Like, there's good papers, amazing papers that show that this is true. So, I think the Canadian Cancer Society and whatnot, they always frown on me when I say this, but because they want to have people like protecting themselves. But the fact of the matter is you can be a non-smoker and get lung cancer. And that's because, the air we breathe has toxins in it. The random chance of your cells making an error, it happens. So, we can do everything we can, but we will never get rid of cancer. We're growing organisms that have cells that grow and divide. To say that we're going to be able to get rid of cancer is impossible. That's saying the only thing we could do to get rid of cancer is prevent aging and prevent our cells from turning over, in which case we would be still an egg. So, yes.
Maybe not a question, but a comment. My sister-in-law, 71, diagnosed with stage 4 breast cancer, double mastectomy.
Yeah.
She's gone through 150 some odd chemo treatments every 3 weeks. Sadly, 2 weeks ago she died. Blood aneurysm, blood clot, which is, I guess, a common thing with chemo. But 10 years is a huge amount of time with stage 4.
It is, yes.
Research works, chemical work.
Yes, they do. Yeah. Sometimes when people, and there's certain, like stage 1 and stage 2 disease, like you see here, some are like 100% survival rates, up to 10 years. Now for breast, especially those ones treated with hormones and the HER2/neu, we consider 10 years not long enough for monitoring survival. So survival curves go out now 25 years. And so, really the needle has changed. When people say that there are no cures for cancer, there are cures for cancer. Certain types of cancers are completely curable. Lymphomas, some of our childhood cancers are curable. And so, I think we've come a long way in the cancer field, most certainly. From my perspective, it's almost impossible to keep up. Every day it's like there's hundreds more papers and the knowledge curve is just astronomical. Somebody asked me when I came in about AI and how that would affect. I'm like, AI is amazing for cancer biology, right? It's going to allow us to go through these piles of data. If anything, we have a data problem. We have so much data coming in. It's about organizing it and figuring out fast ways that we can, make sense of it. And also knowledge sharing, how people communicate. I think now, back in the day, when I started my PhD, it was all about, hey, we've got this discovery and we can't let so-and-so know. Now it's like, okay, so-and-so's known about this for 25 years, like get moving. So it's like, we don't care about sharing knowledge. Like it's about, let's let everybody have the information and let's move things forward. And we find, for myself, it's like everybody and anybody that wants to collaborate and work on a project, let's put all minds together and move it forward. So it's about sharing the data, sharing knowledge, moving things forward.
The 10 years record.
Yeah.
That's fine. Any other questions?
But then, when you say sharing the data and all that, I hear about in the news all the time, is big pharma and how they're protecting proprietary rights that they have. They don't want to share the data. They want to develop the medications that they're gonna generate revenue from. How does that affect what you do?
Yeah, so pharma is a double-edged sword a little bit. We really need pharma, right? And so without pharma, with the first chemotherapy drug back in the 1950s, investing literally millions of dollars to move it forward, it's a super high risk industry. So they put literally millions of dollars on an idea. And so the investment that they put in is huge. It's why biotech and pharma are one of the fastest turnover industries. I think many of the people that work in pharma have a bad reputation in the media and whatnot for what they do. But in fact, it is a business. They do have to make profit and be able to fuel things. So I think it is a little bit of a understanding that we have to be able to have pharma to work with us. I think they've actually become super risk averse because of reputation. And so they used to put a lot more money on research projects. But because society has pushed back on them so much, they've become this insulated thing, which I wish we could actually break down those barriers a bit more, and bring in business people to say, actually pharma requires a huge investment for us to be able to move things forward. Without them, things don't move. Somebody like me, I'm not going to push it through the pharma process. A little grant, like $1.5 million is nowhere near what you need to put the amount of insurance and all that stuff on these patients that are getting brand new types of drugs. It's a very risky business. So I do think we need to kind of change the dialogue. I think people don't understand what pharma does. And we need to be able to work more effectively with them.
I can't remember